Welcome everybody to the HEALEY ALS Platform Trial weekly webinar. I'm just going to give a few minutes for everybody to log on to see the numbers going on, and then we'll launch today. Great. So many of you come weekly, and we really appreciate that. Some of you might be new today. So we're going to give a little bit of background on the HEALEY ALS Platform Trial, but then really focus today's session learning about regimen A, which is the Zilucoplan by UCB. So first, I just want to make some introductions of who's on the webinar today. So from UCB, we have Dr. Petra Duda, Dr. Camille Saye, and then the leads of the regimen A, or the Zilucoplan regimen, are Dr. Christina Fournier from Emory University and Dr. Sabrina Paganoni from the Healey Center in Mass General Hospital. And I'm Merit Cudkowicz, the principal investigator of the platform trial. This initiative, which we launched in July, is the first platform trial in ALS, and it's a way to really accelerate how treatments are developed in people with ALS. And it's really a community effort where we've had funding from the Healey Center at Mass General, many foundations and many partners. And we're working with 4 companies and launching with their treatments. So we've had a series of webinars explaining the science behind each of them. And today our focus is on Zilucoplan. Molly, can I have the next slide, please? So for those of you who are new to the webinar, I want to just briefly explain the idea of a platform trial. So traditionally in drug development in ALS and in other diseases, you test one drug at a time. And so when you have a good idea of something to test in people, you build the infrastructure to run the trial, you analyze the results, and then you take down that infrastructure. And that's an okay approach when you don't have a big pipeline of treatments. But when you do have a big pipeline of treatments, it's actually an inefficient approach. And it's much better to do an approach called a platform trial, which is what we're doing in ALS, where you build the infrastructure for the trial and you test multiple treatments in the same infrastructure, sharing the infrastructure, sharing the placebo data, so that you can really accelerate how you get answers of which treatments work. And it's been shown in cancer trials to accelerate the timeline by about half, so it cuts in half the time to get to effective treatments, and it lowers the cost by a third, and it really increases the number of people who get active treatment. Next slide, please. So we decided that the ALS field was really right for this because there's over 160 companies developing treatments for ALS. There's a rich pipeline. And so we designed this in the following manner. So people come into the platform trial and they're randomly assigned to one of the open treatments. And we currently have 4 treatments in the platform trial. And today we're going to hear about regimen A, Zilucoplan. So people know which regimen that they're assigned to. And then there's another randomization where people are randomized 3-to-1 to get active drug or placebo. So what that means is 75% of people get the active drug and 25% placebo for 24 weeks. And so in this trial, it's 120 people, and each regimen get active drug and 40 get placebo. After the 24 weeks, people have the option to go into what's called an open-label extension phase. And that's where everybody gets the drug. So 100% of the people, for example, in the Zilucoplan trial after 24 weeks could go on open-label extension. And that's for up to a year. And that's enough time so that we will have the results of what we call the double-blind period, that 24 weeks. The efficiency in the platform trial is that when we're going to analyze the results, let's say of the Zilucoplan trial, we'll take the 120 people, the data from them on the active drug, and we'll compare them to the pooled placebo group from each of these arms. And that's where you get the efficiencies in cost and time and how you really increase the number of people who are on active drug. So this is how we designed it, and we're actively enrolling. Next slide. We started last July, despite kind of the pandemic. We're very excited to give some updates on that. So each of the regimens is led by two ALS clinical investigators, and the regimen A, or Zilucoplan, is led by Dr. Sabrina Paganoni and Dr. Christina Fournier, who are here to answer questions along with our colleagues at UCB. Next slide, please. So with that, I'm going to pass the Zoom baton to Dr. Camille Sayegh from UCB, who's going to tell you the science behind Zilucoplan.
Thank you very much, Merit, and good evening, everyone. Thanks for joining. It's a real pleasure for me to be with you today. My name is Camille Sayegh. I head complement biology as well as the microcycles platform at UCB. And I'm going to be describing complement inhibition in amyotrophic lateral sclerosis. Now, in my presentation, I'm going to describe Zilucoplan. Just wanted to remind everyone that this is an investigational drug product that has not been approved for use by the FDA. And therefore, the information I'll be providing is pursuant to a request for scientific information, and therefore should not be, or is not intended to be viewed as medical advice. Let me see if I can change the slide. Perfect. So I'm going to begin with just a very brief introduction to complement and the complement system. I'll provide you more detail on the next slide. But here I just wanted to highlight that the complement system plays an important role in the defense against certain pathogens, for example, certain bacteria, but also has important housekeeping roles, for example, in the clearance of damaged cells. Now we've come to appreciate that in a number of diseases, complement can be dysregulated, can be hyperactivated, or activated in wrong or misactivated in certain anatomical locations, and that has been shown to lead to tissue damage, and in many cases drive disease. And so Zilucoplan inhibits one of the components of the complement system called complement component 5, or C5 for short. Okay, so here I wanted to give you a bit more granularity on the complement system and highlight that there are three main pathways that lead to the activation of complement. And these are initiated by different events or the recognition of different insults or danger signals that the body can perceive. So for example, the classical pathway is typically activated by the recognition of foreign antigen by antibody. And when this forms complexes, that can drive the activation of complement. The lectin pathway is activated by foreign glycoproteins, for example, proteins decorated with certain sugars on the surface of pathogens, whereas the alternative pathway is activated by non-self surfaces, typically that lack the ability to counteract its low level of activation. But all of these pathways can ultimately lead to the activation of C5. And what I mean by activation here is in fact the cleavage of C5, the C5 protein, to generate two bioactive products. One of these is called C5a, and it is a strongly pro-inflammatory peptide. The second product that is generated is called C5b. And C5b, when in close proximity to surface membranes, nucleates the formation of a pore, or if you like, a hole in the cell surface by recruiting a number of other proteins. And we call this the membrane attack complex. And you can imagine that when this happens, for example, on the surface of bacteria, that can help eliminate that invading bacteria. Now, as I've mentioned, we've come to appreciate that dysregulation of any of these pathways can drive disease. And in many cases, this occurs through the activation of C5. And both products, C5a and C5b, and the generation of the membrane attack complex have been associated with disease. So for example, in myasthenia gravis, autoantibodies against components of the neuromuscular junction through the activation of the classical pathway will lead to the membrane attack complex being formed on the postsynaptic membrane and impaired neurotransmission. And it's following this understanding, therefore, that a number of efforts have been put in place to examine the clinical utility of complement inhibitors. And so here I'm going to be describing Zilucoplan. This is a 15 amino acid macrocyclic peptide inhibitor of C5. It is self-administered through subcutaneous injection and functions by binding to C5 and blocking the cleavage of C5 and therefore blocking the generation of both C5a as well as C5b. So moving now to ALS, the neuropathological hallmark of ALS is the loss of both upper and lower motor neurons. And it is this motor neuron degeneration that is associated with the symptoms, many of the symptoms that are seen in this disease, whether we're talking about, for example, difficulty swallowing or the muscle weakness. So what do we know about complement? And here I'll focus particularly on C5 activation in the context of disease. Well, the first insight comes from models of disease. And one of the earliest model of ALS is based on a mutation that was identified in the SOD1 gene in certain patients with familial ALS. So that's the SOD1 G93A mutation. And so when you generate transgenic animals, transgenic mice, meaning mice expressing this particular mutant protein, what you observe is that these mice will develop some of the hallmarks of the disease. And looking closely at examining complement activation, one can appreciate evidence of C5 activation, whether we're talking about increase in C5a, one of the split products, or formation of the membrane attack complex, which I mentioned is the pore at the level of motor end plates. And we also see additional factors being expressed or changed. For example, the C5aR1 receptor is the receptor by which C5a can drive some of its pro-inflammatory effects. And this receptor also is elevated. And so the advantage of animal models is it provides researchers the opportunity to try to intervene and see what biology it could be impacting some of the disease pathophysiology mechanisms that we're studying. And you can do this through different ways. One way is by removing the gene. It's called genetic deletion. So if you delete C5aR1, then this receptor is not expressed anymore in the animal. Or by administration of a small molecule antagonist to C5aR1 to block its function. Both of these approaches in this particular animal model result in extended survival and a slower overall disease progression. Now, how about the picture in patients? And again, I think looking at particularly focusing here on C5 activation products, I'm going to also focus on the membrane attack complex, also shown here on the slide as C5b9. That's referring to the same structure or the pore. And whether you compare or whether you examine upper motor neurons or regions associated with lower motor neurons, there is clear evidence in published studies indicating the activation of the membrane attack complex in the white matter of the spinal thalamic tract, for example, in glial cells of the ventral horn, closely associated to motor neurons. And you see this in patients, and you do not see this in control. And on the right is shown in the lower motor neuron, the detection of this membrane attack complex at motor end plates. Now we also see, and this has been described in a number of studies as well, evidence of complement activations in various biofluids. Here is described the activation of the soluble form of this pore called soluble C5b9. Soluble C5b9 as an indicator of C5 activity overall is seen elevated in the serum of ALS patients versus healthy controls. So these are the data and the materials I wanted to share with you, and I'm looking forward to the discussion and the question. Thank you very much for your attention.
Yeah, thank you very much for sharing the science and the rationale. So now we're open for questions, and so if people could put their questions in the Q&A, we have plenty of time to get to all of them. We have a few that are in already, and so I'll start with those. Well, I'm happy to answer the first one. Maybe I'll go to Sabrina for the first one, which is about Canada. So the question is there anything to do to help participants in the platform trial from Canada when they return home to Canada from America? It's become a nightmare.
Thank you for your question. Yes, I'm aware of that situation because we've been speaking with people from Canada, and I'm aware that recently there have been some changes in the regulations on how to cross the border, independent of citizenship, actually, and that applies also to Canadian citizens who are returning from any foreign country, even if they traveled for medical care. So I know the publicly available regulations, and what we've been discussing with our Canadian patients is that obviously they have to comply with Canadian rules when they go back home. We're happy to write letters of support to support the fact that they were in the US to receive an important medical treatment. Obviously, we cannot guarantee that there will be enough for the participant to get a waiver. I believe that it is possible for Canadian citizens to cross the border and go back. However, as the person who asked the question mentioned, there are very complex regulations, including the need for testing and self-quarantine, and depending on the means of travel, that can imply even costly stay in a hotel to self-quarantine. So again, I'm sorry that there is this current situation, and obviously we're happy to write letters of support, but these are Canadian regulations.
Thank you. Hopefully things will get better as people are getting vaccinated and the world gets closer to normality. But thank you for those of you who are trying to be in the platform in Canada. Camille, there's a couple questions about complement. So can SC5B9 in the serum be used as a biomarker for ALS?
Oh, that's a great question. So soluble C5b9 on its own is not a specific biomarker of ALS. It is seen elevated in a number of complement-driven diseases. So on its own, I wouldn't say it will be a biomarker. It will be interesting to investigate, say, complement activation markers in relation to additional markers, for example, NFL and NFH, and whether those could get us to potential biomarkers of disease. But on its own, it's reflective of complement activation, but not of a particular disease specific.
Another question for you, Camille, is Zilucoplan expected to help damage neurons or just slow down the progression of ALS?
Okay, that's also a very interesting question. And here maybe I can take a step back and describe a bit the membrane attack complex. So I described that the membrane attack complex is a pore, which for example, when it deposits or forms in bacteria, it can lead to their elimination. In nucleated cells, such as neuron, the deposition of the membrane attack complex, so nucleated cells have their own means to push back against activation of complement and to try to neutralize it. And so often what happens, there is two stages. There is one stage that is sublytic, meaning the membrane attack complex sits on the cell and causes a stress to the cell, but it is not leading to its death. But if this process continues, it can ultimately lead to cell death. So I would say one can conceive that in a situation where the sublytic MAC is impairing cell function, that blocking that process is going to be beneficial. Obviously, if the cell has been pushed to die, then you're only preventing or slowing disease. So depending on the context, you could reverse damage, and you can certainly hope that we can stop disease progression.
So that's something that definitely we'll learn from this study. So that's
yes.
So there, I don't know if Christina or Sabrina want to take this one. I think I might have misspoken, but there's someone who was paying close attention who said, I remember several months ago, the open label extension for Zilucoplan was not limited 1 to 1 year. Has this changed?
Christina, do you want to go or?
Go ahead, Sabrina.
Yeah, so essentially the open label extension could be longer than 1 year. So basically the extension can be different for each regimen, and you're correct. For regimen A or the Zilucoplan regimen is longer than 1 year. It's actually written as if it can continue sort of, for sort of an indefinite period of time. But obviously the assumption is that before then we would know the results of the drug, and so the plan is to basically extend it, again, long term if the results are positive. But as of right now, there's no end date. Yeah, you're right.
Thanks. I want to say that there are some questions about a different drug, neuron, and I'm going to leave those for the end. Just if we have time, we'll get to them. We'll hopefully have another webinar on that. But I do want to focus this one on Zilucoplan and the platform trial. But I didn't want someone to think I was ignoring them. So there's another question about the blood levels. So for Camille, are C5b9 levels being evaluated in participants in the Zilucoplan placebo arms?
Yes, it is. We always evaluate complement activation and C5b9 as a marker of complement activation and how that is changed with treatment. So with Zilucoplan. So this is something that we look for.
So Christina, maybe the next one for you. So someone's mentioning that their wife is in the trial and is in regimen A, but only for 2 weeks. They're 2 weeks from the end of the 6-month period. We're wondering if you observed any promising signs in the participants. I thought maybe you could just talk about the double blind and how trials work.
Yeah, so we also are very excited, but we are blinded still, which means that we don't know who is receiving drug and who is receiving placebo. So we do not know yet how well the drug is working. So that's required for science to be really objective and to make sure that we're looking at it fairly and that we don't have any biases as we're treating patients in the trial. So we don't know yet is the answer, but we're hopeful.
Kind of a related question, maybe Camille, you can answer it. Can you comment further on the efficacy and safety results in clinical trials to date in ALS and other diseases?
So I can comment perhaps on the one that we've published, and maybe I'll invite Petra Duda, who's overseeing our clinical studies as well, if she wanted to add anything. And that is the study that we've done in myasthenia gravis. Phase 2 trial, and I've kind of alluded to it in one of my slides where that phase 2 trial concluded positively. This is a trial in a subset of patients with myasthenia gravis that have particular autoantibodies. I was matching that against the neuromuscular junction. And in that small phase 2 trial, we saw positive data indicating improved overall primary and secondary endpoints that were being addressed in this particular study. Petra, anything you wanted to add?
Yes, thank you, Camille. And of course, that small study led us to continue observations and continue into a phase 3 study in GMG. And it also showed us that the drug was well tolerated in that phase 2 study in GMG patients. And there were no major safety signal. As you all know, there is one major potential risk associated with complement C5 inhibition, and that is Neisseria infection. And we're taking all the precautions that we can to help prevent that, of course, in this trial as well as in other trials as well.
I'll just add that this is the first trial in ALS, so we don't have any prior.
We don't have the data.
So there's a question about how your drug is different from Ultomiris and the Alexion champion trial. I don't know if you can comment on that.
Well, I can comment maybe at the high level, which is the drugs that are being referenced by Ultomiris fall into a different class of modality, if you like, which are monoclonal antibodies. And so both these monoclonal anti-C5 as well as Zilucoplan mechanistically are doing something similar, which is blocking C5 activation and therefore blocking the generation of C5a and C5b. So mechanistically, at the high level, it is similar. Now, obviously, these agents have different mechanisms of actions in the sense of how they bind. Zilucoplan, being a small peptide, also is administered differently through, for example, subcutaneous daily injection. And we believe, based on its size from the data we've reported, that it has the potential to distribute in the body in a way that's different than what you see with, for example, monoclonal antibodies. So overall, similar biology, but the modalities and the type of drug has differences that one needs to examine and study as well.
Another question again for you, Camille, which is that you mentioned that Zilucoplan blocks C5a and b, and in the transgenic mice, it resulted in increased survival. They were wondering whether there were any functional benefits as well in the animal model.
Oh yeah, great. So let me first clarify. That's a very good question, and let me first clarify that in that animal model, Zilucoplan was not used. What was used is antagonist, so a tool compound that people use in research to block one function, which is the C5a receptor. So in that particular animal model, C5b was not blocked, and therefore formation of the membrane attack complex was not impeded. In these animal models, what they've seen beyond survival is also, as I mentioned, slower disease progression, and that refers, at least in the animal model, to the animals retaining, for example, muscle strength longer as the disease progressed. There was also markers of inflammation that were looked at, and these were also changed by blocking the C5AR receptor.
I wanted to maybe turn to Sabrina and Christina to maybe answer or give your thoughts about this question about what happens after you're done with the 24 weeks and you can go into the open label. There's a question about whether you can find out at that point what you were assigned to, active drug or placebo, and if not, when would you find out? Christina, you want to go first?
Yeah, go ahead, Christina.
Oh, so it's not right away that we're able to tell people, because after everyone's completed with their 6 months, then the data has to be analyzed, and we have to have that data analysis with everyone still being blinded so that we're sure that those results are really unbiased and accurate. And so it's some period of time after all of the study participants have completed their 6 months, and then the data has been analyzed. It's hard to put an exact date on that because it'll depend on when recruitment is complete and when the data analysis is complete, but there is a bit of a delay because the results have to be analyzed first.
Yeah, absolutely. And the open label extension is really an integral part of the trial. So essentially, the open label extension is very important for several reasons. One, to give access to the active drug to the participants who participated in the placebo control period, but also to capture in a systematic way and unbiased way long-term safety and survival and long-term outcomes. So essentially, it's important for us to, again, continue the research, but then certainly after the final results of the double blind are done and all the data has been completed and we get, again, all the main results available, at that point, it's possible to unblind and share the treatment assignment with everyone.
When we met with the FDA about the platform trial, which again, they were very helpful, they really did make this point of asking us to keep people blinded when they moved over to open label of what they were on before, because it is a way to get, again, additional data. And I think the studies Sabrina led before, the AMLEC study, actually is a very good example of how much more you can learn from that, and that helps the field and helps get towards the goal of getting drugs approved for people like us. There's a couple of questions around the bacteria that I think are really good that I want maybe Camille and Patrick to answer. So the question is whether you think complement inhibitors will work best in people whose ALS was caused by bacteria and viruses. So what's the kind of link between that?
Yeah, I think that's a great question, and I think that's getting to what's driving pathology and what's driving complement activation. And I can say that at this stage, this is still something that we need to understand better. And I see another question following to that one on the endogenous, so HERV-K, so endogenous retroviral activation. I mean, this is a great question because again, if something like this would occur, then you can think about a mechanism where, say, antibodies will be recognizing these and triggering activation of complement.
So maybe I'll add that. So we're not, there's no data that this only works with infection. So,
right.
So as Camille is saying, that we don't know what's driving the complement activation. And we are banking blood from everybody so that we can go back and look for things like the question on HERV-K.
And I would add, if you look at the studies that I referred to in patients on, say, MAC activation, this was, I mean, these were small studies, say 16 patients or so forth, but activation was seen through in every patient. So if it was restricted to a small subset, then you would investigate what could be driving that, and maybe it's infection agents or not. But it appears to be, it was observed across patients, which made me think it's something more generalizable that's driving it.
So Petra, someone didn't quite catch the infection risk. Maybe if you could repeat the name, but also talk about what we're doing to prevent that.
Happy to. So the risk is again, Neisseria infection. It's a bacterium that can mostly cause meningitis, but also a venereal disease. And those are two different bacteria. And what we're doing is actually vaccinating every patient who is coming into the trial with the available vaccines. One of them is meningococcus B serotype, and the other one is against ACWY serotype, so multiple different serotypes of the same bacterium. Every patient gets both of these vaccines, and they need to maintain vaccination during the trial as well. And in addition, we're providing every patient a patient safety card as well, which explains in detail the symptoms that these patients can have when the meningococcus attacks them. And it also directs the patient to go directly to the physician, to an emergency room, because it's time critical. Once these symptoms occur, you have to go to the emergency room and get antibiotic treatment as quickly as possible. And this is all explained. And in addition, when the patients get randomized to this trial, to this regimen, they will also get an additional explanation from the investigator as well.
Again, the importance is knowing about the risk of then being able to give the vaccine to people. So it's not a great risk. So I don't want people to leave being worried about it, but it's important to know the risks so they can mitigate them. So maybe Sabrina, if you could answer the question, if the results are positive, do you anticipate the injections to be needed on a regular basis or just one time?
What we're testing is a daily injection regimen. So the assumption would be that if it is positive, that would be the ideal regimen for patients to continue on. But so that's a hypothesis that we're testing in this trial.
So Christina, there's a question about what happens when you move to open label extension. What is the visit follow-up schedule?
So there are still visits, but it's not as frequent, I believe, in the open label phase. So I don't have the exact schedule memorized, I must admit, but the requirements for in-person visits do decrease a little bit in frequency. So there still is some safety monitoring and some ongoing questionnaires and things like that, but it's not as frequent as it is in that 6-month randomized phase.
Yeah, definitely. The goal is to provide access while reducing sort of the need to again attend all the visits and do all the procedures. I do want to mention that at the beginning of the open label extension, the schedule is quite similar to the placebo control period, because we need to ensure that we can monitor the safety of all participants, but especially those who were previously on placebo, because they would be exposed to the drug for the first time during the beginning of the open label extension. So that will be for the first 24 weeks or so. And after that, the frequency goes down to less frequent visits. In general, even during the beginning of the open label extension, there's fewer outcomes though. So certainly the visits become shorter.
So there's a question about, from your prior myasthenia research in Zilucoplan, how long did it take for the C5b9 levels to drop? And is the dosing of Zilucoplan the same in myasthenia trial as it is in the ALS trials?
So I'll take the first question, and then I'll hand over to Camille. So the dosing regimen is exactly the same as it was in the GMG trial. So we're expecting the same safety profile or similar safety profile in ALS patients here as well.
And I would add that all our work to date shows that following Zilucoplan injection, the inhibition of C5 activation is very rapid. You see it within hours, and you see therefore the decrease of soluble C5b9 happening very quickly as well.
So a related question is when it's injected, does it have to get into the brain or does it work peripherally or both?
We don't know, but as you've heard from Camille, there are certainly points of attack for a potential mechanism, both peripherally as well as in the CNS. And I don't know whether you want to say something in addition, Camille.
No, I think you said it best, and the data I was referring to in terms of inhibition being very rapid, that's in the periphery where we studied it the best. The CNS is something we are investigating as well.
Yeah, I would say, I mean, you did show us that you see the complement activation both in the muscle and in the brain.
Right.
So Sabrina, there's kind of another question around the transition to open label that I was hoping maybe you could answer, which is that people with ALS don't have time to wait until they know if they were on active drug or placebo. So if they were on regimen A, or really if they're on any of the regimens, and they don't feel like they've had benefit, what options do they have other than open label for Zilucoplan?
Yeah, I would say that, you know, this is a great question. It's also a complex question that brings up different topics within that. So the one thing I would say is that, you know, in the past, there was no option for open label extension for the vast majority of ALS trials. And now more recently, and especially with this trial, it's an addition in the spirit of giving people access to the drug while we wait for the results so that we know if the drug works in the population. On an individual level, it is true that that creates the need to make a decision before learning the results of the trial. And I would say that, you know, it can be difficult for an individual to know if there was a response, just thinking about one's own experience, because there are things that can affect that. And so I think that before concluding on an individual basis that the drug works or not, it is really important to get the results for the entire population, because, you know, you obviously don't want to continue a drug that doesn't work, but you also don't want to not get access to a drug that could potentially work, even if perhaps there were other factors that made it difficult to really be able to tell on an individual basis if the drug worked or not. I don't know if you want to add anything else, but I think there are multiple layers here.
Yeah, I think again, with the feedback from our patient advisory group and a lot of people, we designed this to optimally give people as many choices as possible, so people can go in the open label, they can decide not to, they can re-enroll in another regimen and go to another trial. And it's really a discussion with you and your neurologist and your team who know you well, just to go through all the options. And it can take time, those are complicated discussions, but they're really important to have. And you know, hopefully if you're in the study that you have that relationship with the team there, that you can have those discussions. There's a question about whether there's any plan to include Zilucoplan in an expanded access program. And I don't know if Petra, you want to answer that, or Sabrina? You're
on mute.
It's amazing how long it takes to unmute yourself when you're nervous about trying to do it. Anyways, so we're currently focusing mostly on the extension study in terms of providing access to patients. And as you know, we're in discussion about potential early access, but that is certainly something that we're currently in discussion with you and not yet ready for prime time with.
So there's a question unrelated to Zilucoplan of the platform trial, and maybe turn this to Christina and Sabrina, which is really about any new treatments for the familial form of ALS caused by C9 mutations.
Christina, go ahead.
Oh, so there are currently gene therapy trials specifically for C9 patients. Those are still in early phases, so we don't have results yet. And those are happening separate from the platform trial because the platform trial really has to enroll patients with the same criteria. So gene therapy trials for specific genetic mutations didn't really fit into the framework of platform. So those are happening kind of separate from platform. No results yet that I've seen, but there are several that are ongoing.
Yeah, and I would say that it's such a promising time especially for C9 research, because there are many other companies that are developing new ideas and new compounds in addition to the gene therapy or antisense oligonucleotides tools. So that's certainly something to continue to discuss, and hopefully we'll see more trials in the near future.
We are starting actually discussions with the ALS Consortium about a platform trial for C9 only. Because I think there's starting to be enough companies in that space that it's going to start to make sense to do this similar approach just for people with that form of ALS. Good. I don't. Oh, okay. So so we have two questions that are unrelated to this, are really about brainstorm and neuron. And I think I want to say that I do think that the company and the investigators involved in that will have a webinar just about that at some point to show all the data. And they're also planning to publish the paper. I know that everybody read that the FDA has declined the company submitting for. And I think the reason is what was publicly stated, is that the trial was negative. But I think the most important thing is to get the data out so everyone can see it, and to have a webinar that's focused just on that one, so that the company and the people seeing the data can really answer those questions. I know everybody was disappointed about that. And every time there's a negative study, it's enormously disappointing. But I remember one of my first trials that was negative to pyramate, and I got a couple of letters from, in particular one from one of my patients who said, "Okay, what's next? What else are you going to sign me up? Don't give up." And so I keep that next to my computer. We have to keep going and keep understanding the science and keep going with better and better drugs. And that's what the platform trial is about. So there's a question about how many participants are enrolled in platform regimen A today. I'm going to go to Sabrina or maybe Lindsay, who are on top of all these numbers.
Yeah, I'm sure Lindsay will know better than me, but it's about 80, 70 to 80 that have been assigned, and there is a 2-week wait period to get the first vaccination dose, but it's basically equally distributed among the first 3 regimens that we are in the 70 to 80 patient area. Yeah.
So we're about halfway done for the first 3 regimens, so we're very excited about that. I think, again, it's a difficult time for everyone because of the pandemic, but this trial is really enrolling well, and thanks to so many of you and other people who are interested in being part of this study. And the exciting thing is we'll have answers to all 3 at almost about the same time. I don't see any other questions. I don't know if in the last couple of minutes, if maybe I'll go to Camille first, if you have any other comments you'd like to share.
Merit, was this for me?
Yes, yeah, if
okay.
any last words, we'll ask everybody.
Look, I think this is, from my end, the first time I do a venue like this. I'm really appreciative of the attention. I'm appreciative of the feedback, the questions. A lot of them are very good questions. And for me as a scientist, it's always motivating to get to hear from patients, and that it helps us, in really drug discovery is difficult, and it gives us the wind in our sail and realizing the urgency and the task we have to do. So from my end, this is a venue that I think is very rewarding as a scientist.
Thank you. Petra, any last words?
It's a hard one to follow, but I'll try. So I mean, it's really been humbling to see how you, Merit, have been able, together with Sabrina, to bring this platform trial to life. And I would like to also express our gratitude to all the patients that you have been able to motivate and work with to participate in a trial, because it takes courage to participate in a trial. You don't know what you're going to get, right? And you've come together as a community, helping to develop drugs for yourself, for everybody else who has this disease. And I think this is really a great moment here.
Thank you. And then I do want to hear last words from Christina and Sabrina, but we did get another question. I don't know, Sabrina, if you want to answer this one. This is how many in each regimen are we targeting, and what happens if we don't reach full capacity?
Yeah, we're targeting 160 per regimen. And that's a great question, actually, because in the past, in general, in medicine, many trials didn't reach capacity, but I don't think we're going to have this problem here, because there's really really great collaboration with the patient community, and we are so grateful that you're interested in participating, as well as great collaboration with the investigators, the sites, and the industry partners. And I think really the enrollment has been exceeding expectations in general, even for non-pandemic times, and it's quite remarkable to see that there is such strong participation despite the pandemic. So I think really we will finish enrolling, and we will be able to get you answers. In fact, we're gonna get you answers faster than we were able to ever able to do before even, you know, we had the platform trial. So thank you for that.
Thank you. Christina, any last words?
No, I just want to echo our thanks to the patients who I think have been, you know, so trailblazing in this and so motivated to kind of be a part of finding treatments for ALS. And this trial has been really exciting. It's kind of enrolled at lightning speed, and it's been innovating in a lot of ways. So I think, you know, the patients have made this better at every step of the way with their input, and we really appreciate that.
Thank you to all the panelists and all the attendees. We will put up the recording on our website and hope to see many of you back next week for our weekly platform webinar. Thank you.
Thank you.
Thank you.